Reception is the target cell's detection of a signaling molecule coming from outside the cell. The signaling molecule binds to a receptor protein located at the cell's surface. Transduction is a step or series of steps that converts the signal to a form that can bring about a specific cellular response. In the third stage, transduced signal finally triggers a specific cellular response. The response may be almost any imaginable cellular activity, such as catalysis by an enzyme, rearrangement of the cytoskeleton, or activation of specific genes in the nucleus.
Enzymes are biological molecules, typically proteins, that speed up the rate of virtually all of the chemical reactions that take place within cells. All the work being done inside any cell is being done by enzymes. The purpose of an enzyme in a cell is to allow the cell to carry out chemical reactions very quickly. In any chemical reaction, a substrate is converted into a product. Enzymes are made from amino acids. Two molecules join together to make a larger one. The substrate reactant moves towards the enzyme's active site. The chemical reaction is triggered by the enzyme. The enzyme releases the products. In the end, the enzyme is by itself again.
For a signaling molecule to undergo the reception process, it first must be recognized or detected by its target cells, just like a lock and a key, hence the name lock and key theory. To think of as two puzzle pieces. Not all puzzle pieces can fit together. Only the puzzle pieces that have similar size can fit together. The signaling molecule is called a ligand. This ligand is a smaller molecule that attaches itself to a larger molecule, changing its shape, sort of like a key twisting the lock to open it. Once it's attached and changes its shape, it enables it to interact with other molecules.
Transport is whenever molecules or ions move against their concentration gradient across a cell membrane, but requires the use of ATP or energy. So one way that active transport occurs is through membrane pumps, as shown here in the sodium-potassium pump. So outside of the cell, there is a higher concentration of sodium ions, and inside the cell, there is a higher concentration of potassium ions. Naturally, the cell wants less sodium ions and more potassium ions, so the ions have to move against their concentration gradients. Three sodium ions will bind to a transport protein, as shown here. With ATP, a phosphate breaks off, binding to the transport protein, which causes the transport protein to change shape. This releases the sodium ions into their higher concentration gradient. And then while that happens, two potassium ions move into the transport protein. The phosphate group from the ATP breaks off, changing the transport protein back to its original shape, releasing the potassium ions into their higher concentration gradient. Another form of active transport is bulk transport, which occurs through endocytosis and exocytosis. Endocytosis is when large molecules are taken into the cell, and exocytosis is when molecules are pushed out. Endocytosis occurs through phagocytosis, which is cell engulfing, in which a large molecule, like this highlighter, is wrapped around by the phospholipids with energy. It is engulfed and taken into the cell. Penocytosis is another form in which it is like cell drinking, where solutes are similarly wrapped around by the phospholipid bilayer, taken in to the cell. And receptor-mediated endocytosis occurs when there are specialized receptors that take in certain molecules, wrapping them around and taking them into the cell. Exocytosis is the opposite of endocytosis, in which a vesicle inside the cell pushes up against the phospholipid bilayer. It opens up, releasing the chemicals inside out of the cell. Secondary active transport is like primary active transport, in which it requires energy but does not need energy directly from ATP like primary does. So secondary active transport gets energy from the ion moving down its concentration gradient. But in secondary active transport, it is coupled with a molecule moving against its concentration gradient. So as seen here, there's ions, a higher concentration of ions inside the cell, and a higher concentration of molecules outside the cell. So the higher concentration of ions is naturally going to go down through the cotransporter to the lower concentration gradient. But coupled along is going to be a molecule, which is going to use the energy from the ion to go up the concentration gradient while they both move outside of the cell.